everyone, welcome back to another video, and today I will be explaining the cell cycle. And this is explained easily. So what is the cell cycle and mitosis? The cell cycle is the process a cell goes through to grow and divide into two new cells. Cells divide when they need to grow, repair damaged tissue, or replace aging and dead tissues and dead cells. In the human body, this continuous process is called mitosis. So the cell cycle is usually pictured in a circular diagram split into 4 equal parts. So for the first phase, you got G1, then S phase or synthesis phase, G2, and then you got the M phase. G1, synthesis, and G2 phase altogether are known as the interphase. In addition, M phase has a lot of phases too. M phase consists of prophase as the first phase, metaphase as the second phase, anaphase as the third phase, telophase as the fourth phase, and finally cytokinesis. Now there are a lot of objects in the cell that you have to understand. These dots on opposite sides of the cell or opposite poles of the cell are called centrioles. These dashed lines or stringy-like things are called the spindle fibers. These chromosomes that look like a V are called sister chromatids. The dot in the center of the chromosome is called a centromere. This region contains a protein structure called kinetochores where the spindle fibers will connect. And finally, in this nuclear bound membrane, it contains DNA. So what is interphase and M phase? Interphase is when the cell grows and prepares to divide, and it occurs in most of the cell cycle. M phase is when the cell actually divides. So what is prophase and what does it do? Prophase is the first stage of the M phase. Chromosomes condense and become visible. The nuclear membrane breaks down. Centrioles move to opposite sides of the cell. Before cell division, they duplicate. Spindle fibers begin forming. There are some key terms to know before moving on to the other phases. Chromosomes are things that carry DNA. Sister chromatids are identical copies of DNA that are halved, especially the ones I mentioned earlier that look like a V. A centromere is the object at the center of a chromosome that holds the sister chromatids together. The spindle fibers is also known as the spindle fibers attachment site. A kinetochore is a protein where spindle fibers attach. Centrioles are an organelle that organizes spindle fibers and organizes the cell in preparation for cell division. There are 2 of them in each pole while preparing to divide. And lastly, spindle fibers are string-like things that attach to the centrioles and the kinetochores, and they also attach to the centrioles. Next, what is metaphase and what does it do? In metaphase, the chromosomes line up in the middle of the cell. It is the second stage of M phase. So when chromosomes line up in the middle of their cell, they are preparing to be pulled to opposite poles by the spindle fibers. So as you can see in this image down here, the string-like things or or as you see right now, the dashed lines indicate which direction they are going to go. Next, what is anaphase and what does it do? Anaphase is when the sister chromatids get separated and pulled to opposite poles. It's the third phase of M phase. As you can see in this picture on the right, not only did the cell extend in its height, but the sister chromatids, or these V-shaped looking chromosomes, get pulled closer to the centrioles. So what is telophase and what does it do? Telophase forms new nuclei and chromosomes uncoil. A tip when thinking of telophase. When thinking of telophase, think of it as if you're trying to split clay into two parts with your bare hands. It will look like a peanut. In the cell image on the right, the two identical daughter cells are beginning to split into two equal parts rather than just being connected together the whole time. And finally, what is cytokinesis and what does it do? Cytokinesis is the final process of cell division that splits the cytoplasm of a single eukaryotic cell into two distinct daughter cells. The cell membrane pinches in, and two identical daughter cells are produced. In the image below, it shows that the two daughter cells have officially separated from each other and they are no longer connected with that middle part. So if we go back to our diagram from telophase, we can see that during telophase, it's about to split, but it didn't split. So when you think of cytokinesis, think of it as if the two cells bisected each other. And when they bisect in the middle, they have to repair their membrane. Just like when your body gets a cut or an incision, its membrane naturally has to seal itself. Remember, the goal of mitosis is for repair and refinement. So that's it for the end of the video. Thank you for watching, and I'll see you in the next video.